There is a new link where we'll be streaming from. So we can now start without further ado. Just a little bit of housekeeping. You can send any questions you have throughout the talk on the YouTube chat. And at the end of the talk, this will be answered by the speaker. And just a friendly reminder that we have the BSGCT annual conference coming on the 20th of June at Royal Holloway, University of London, and registrations are now open. So now I'm gonna stop sharing my slides and my video, and I'm gonna, before, give a brief introduction to our speaker today. Alberto, thank you for accepting the invitation to be our guest speaker. Dr. Alberto Malerba holds a PhD in biotechnology awarded by University of Padua. He joined the gene therapy laboratory at Royal Holloway, University of London in 2007 to work as a postdoctoral scientist. This lab was then directed by Professor George Dickson. And since then, Alberto has worked on the development of novel gene therapy agents and antisense therapeutics for the treatment of rare muscle diseases. He initially focused on accelerating the clinical translation of exon skipping for Duchenne muscular dystrophy. Afterwards, as a research officer and project manager of the Dickson lab, he collaborated on the development of second-generation gene therapy products for DMD that are currently on clinical trials. He has also worked on new drugs and gene therapy approaches for the treatment of Oculopharyngeal Muscular Dystrophy, OPMD, a rare muscle proteinopathy. In this context, he has developed a gene therapy AAV vector in collaboration with Benitec Biopharma, and the vector has recently entered our first in-human phase 1/2 clinical trial in the US. He is lecturer since 2022, and he is currently leading the gene medicine laboratory for rare diseases at Royal Holloway. So, Alberto, thank you for joining us today.
Thank you. Thank you, Rebecca, and thank you to the organizer for inviting me. So I will share my my screen. I hope. Able to do this. Okay. Okay, so let's put down this. Okay, so yeah, so my name is Alberto Malerba. So basically, I'm working at in Negam, where we have a laboratory at Royal Holloway University. It's a beautiful campus here. And yes, in my lab, we are developing gene therapy and I would say antisense therapeutic treatments for neuromuscular conditions. So I think, okay. So this is a summary of the major muscular dystrophies, and my work is in particular focused on developing treatment for 3 diseases. I will do this actually because I think it's better. So Duchenne muscular dystrophy and Becker, the allelic form Becker muscular dystrophy as well. The FSHD, so facioscapulohumeral muscular dystrophy, and oculopharyngeal muscular dystrophy. They are diseases with very difficult names, and they are also pretty complex as diseases. But basically, today I'm going to focus on 2 of them, Duchenne muscular dystrophy in particular, and oculopharyngeal muscular dystrophy. So why these 2 diseases in particular? They are very different. Duchenne muscular dystrophy is an X-linked recessive disease. It's a loss of function disease, which basically means that you have to provide the missing gene because there is a mutation, so you don't have the gene working, you don't have the protein there, so you want to rescue the mutation. So this is the kind of approach you would do. It affects young boys, and the mutation you have in the gene are generally out of frame mutations, so you don't have the protein at the end. Oculopharyngeal muscular dystrophy is a different disease, an autosomal dominant disease. So this is due to basically a triplet expansion in a gene that codes for a protein that has a lot of different roles. And it's a loss and gain of function disease because the protein due to the mutation, will aggregate. So it aggregates. So you have that the aggregates can actually have a detrimental effect in the cells, but also you have a loss of function because there's less of the working protein there. This is an aggregopathy. It affects mainly a mature population, so people that are in the 40, 50, 60 years of age. And it is a trinucleotide repeat. So it's really a completely different disease. And this is probably why I focus on these two, mainly on these two diseases, why my interest is in these two diseases. So what they have in common, these two diseases? First of all, of course, they are muscle conditions, both of them, and we are working muscle. They can be both approaches using gene therapy applications, and this, of course, is very interesting for us. And also, they can be considered models, so sort of prototype when you want to develop treatments. So you have a loss of function disease, you have a gain of function disease, so you can actually use them as an example of what to explore therapies for other treatments, for other diseases as well. So I will start with Duchenne muscular dystrophy. This is, as I said, an X-linked recessive disease. It's the most frequent muscular dystrophy, and basically it affects children. The mutation is in a gene that code a protein. It's called dystrophin. You have here, there's a type of a muscle. Basically, you have a muscle. You remember that the muscle, the myofibers, so the muscle cells, are very long. They are syncytia, multisyncytia, made by many nuclei of the same cell. And around the cell, we have a sarcolemma, which is the membrane of the cell, where there are these proteins here that are called dystrophin-associated protein complex. So they are proteins that are very important in the cell. And if these proteins are missing, is a massive issue for the muscle cell. These proteins are connected to the cytoplasmic actin through this very long protein, it's called dystrophin. And again, if dystrophin is missing, these proteins will be missing. But also, if dystrophin is missing, the myofiber collapse. So what you have is that the muscle cells will not resist to all the insult that the muscle have during the life. So basically, the muscle itself will collapse, and you will have a replacement of myofiber with fibrogenic adipogenic tissue. So this is an example of immunostaining for dystrophin, where you see the protein at the sarcolemma here in black, and of course in the Duchenne muscular dystrophy muscle you don't have that. As I said, the disease affects children. It's a very severe disease. Children, unfortunately, go to a wheelchair early in life, and the patients with the disease die when they are in the 20 to 30 years of age, mainly due to cardiomyopathy and respiratory failure. I have to say that now there are some improvement because there's a possibility to have ventilation, there's physiotherapy, there's the use of steroids that actually can improve a little bit, but in reality there's no cure for this disease. So dystrophin, I said dystrophin protein is very important. This is a schematic of a dystrophin protein. So this is basically made by an N-terminal domain, which bind the cytoplasmic actin, a C-terminal domain, where there is this region, which is called the cysteine-rich region, that is bound to the beta-dystroglycan, that is one of the proteins of the dystrophin-associated protein complex. So basically here links to the cytoplasma, here linked to the sarcolemma. And in the middle, you have a very long spacer, which is made by 24 spectrin-like repeats. And spectrin is a sort of structural protein, and this is divided by 4 hinges, which give flexibility to this protein. So we have 2 allelic forms of the disease, depending on the type of mutation in the gene. So if we have out-of-frame mutation, we will have basically the formation of a stop codon at a certain point. And the fact is that we have a non-functional protein, and we have Duchenne muscular dystrophy, so we have no protein in the muscle. If instead we have a deletion, for example, that keep the reading frame in frame, we have deletion that are in-frame deletion, and so we'll have a protein that is missing of part of the internal domain, but still the N-terminal and the C-terminal part will be there. So we will have a shorter but still potentially partially working protein, and the phenotype is generally less severe. Again, here it depends on the length of the deletion, really. But if the deletion is not too long, you potentially have a relatively mild phenotype, and we have a Becker muscular dystrophy phenotype. I put here this table, but basically this says just that generally with the Becker muscular dystrophy, for example, you don't have respiratory insufficiency, you have much less people that go to a wheelchair, so it's definitely a less severe condition. And this paper came out in the 1990. It was from Key Davis group. It was really important because it showed that there are cases of people with Becker muscular dystrophy that are really mild, and where you have a large deletion of the protein. And so, of course, scientists start to say, okay, so let's try to convert a Duchenne muscular dystrophy to a Becker muscular dystrophy, if that is possible, in order to treat the disease. And so there were cellular models and mouse model developed to try to resolve the disease. And basically, two genetic approaches were identified mainly, and now they are in clinic, and they are actually already FDA approved. One is called exon skipping, and the other one is called AAV gene therapy. So, and I was lucky enough to work in both in my life. So years ago, we did a number of experiments in the mouse model. This was a publication that came out actually many years ago, and in the MDX mouse. And in this mouse model, we have a mutation in the exon 23. There's a formation of a stop codon in the exon 23. So the dystrophin is mutated and then is truncated, basically. Exon skipping is based on delivering an antisense oligonucleotide that can mask specific sequences into an exon. And so in the pre-RNA messenger, so that when you have the splicing, basically the splicing machinery will not recognize that exon as an exon, but it will think is an intron. And this is great because then it can skip that exon when it's the time of having the formation of the final RNA messenger. And the fact that you can potentially delete some exons from the final transcript, providing specific antisense oligonucleotide. This is done in the case of the MDX mouse, for example, here. And if you do that, you see that you can restore dystrophin expression. This was an immunostaining. In this particular paper, we show that if you provide weekly, in this case, dose of a chronic injection of oligo is better than actually giving the whole amount in a single time. And this was also corroborated by another study, which was a very long study. This lasted for more than a year. So we did monthly injection of 2 different doses of antisense oligonucleotides for a year, and we saw a nice increase in dystrophin expression, a decrease in creatine kinase in the mice, showing basically creatine kinase is an enzyme that is released by the muscle when there is damage. So you really want to decrease that to see an improvement in the pathology. And there was an improvement in the forelimb grip strength, so the strength that the mice have in their forelimbs, and also an improvement in a measurement called resistance to eccentric contraction, which is really important, and basically is a number of damage you make to the muscle, and you measure the maximal force that the muscle can anyway provide, even if it is damaged by these consecutive damages. And you see that both doses actually rescued the muscle force compared to the MDX. So there were these data coming, we're talking about 10, 15 years ago, and many more from other groups. And there were companies interested to the pathology, of course, and one in particular was Sarepta Therapeutics. that started to work on the clinical trials with patients and developing this exon 51 skipping. So this is an oligo that skipped exon 51 in particular. And I don't go into the details. This is basically a 6-minute walking test where you measure the how long a patient can walk in 6 minutes. And in this particular study, there were 2 cohorts of people involved. One was injected from the beginning with antisense oligonucleotides. Another cohort was initially provide placebo, but then after 24 weeks, they started to have the antisense oligonucleotide as well. And you see that in both cases, when the dystrophin starts to kick in, dystrophin expression starts to kick in, you have actually a stabilization of the pathology. And this clinical trial and more from the same company and from other companies basically provide all the proofs necessary to approve these products in clinic. Now we have 4 exon skipping strategies approved for 3 different exons, exon 51, 45, and exon 53. So in the lab where I was working, George Dickson and Linda Popplewell designed the sequence that is now this BioNDis 53 exon for the exon 53. And as you say, each exon potentially can treat a cohort of patients. So this is not a therapy for every patient. You really target a specific cohort of patients with this. But Sarepta Therapeutics also is developing antisense oligonucleotides for other exons as well, and also in theory system to improve the efficacy of these antisense oligos. So using cell-penetrating peptides to try to improve the permeability of this antisense oligo. But I have to say that recently we had the bad news that these clinical trials were stopped, and mainly due to toxicity in the kidney. So cell-penetrating peptide is extremely effective in preclinical study for sure, in the animal models definitely, but we know they are associated to toxicity. So the problem is to find a peptide that works with and it is not toxic. That is really the crucial point. So there are limitations of the antisense oligo, of course, inexpensive treatment, and they have a limited penetration, as I said. For example, there's no effect in the heart or in the brain. And in the heart, in particular, we studied again many years ago. We saw that in the heart, in the cardiac muscle of the mice that were treated for a year with the antisense oligonucleotide, actually, at the end of the treatment, we were finding more fibrotic tissue. For example, we are finding more damage into the heart, suggesting that with this therapy, you create individuals, I mean, in our case, they might, but in a clinical trial, boys that are potentially more mobile, more able to walk, potentially to run, but where the heart is not really treated. So there's really needed to try to get some better system, better treatment. So potentially with cell-penetrating peptides, as I mentioned, or trying to develop new chemistries, or even adding other treatments. And I would say that we focus mainly on this 3rd point here. And for example, we developed a myostatin knockdown using exon skipping as well. So myostatin is a negative regulator of muscle growth. So basically, if it is upregulated, you will have muscle atrophy. If it is downregulated, you will have muscle hypertrophy. So it's one of the factors that are really important to keep the size of the muscle the correct size. And you can downregulate in many different things. Myostatin antibodies have been developed to do that. We developed an exon skipping. Basically, in this case, we have exon 1 and exon 3 that are out of frame. So if exon 2 is not present, we'll have a formation of a stop codon at exon 3. And so designing antisense oligonucleotides targeting exon 2, we could actually dysregulate the formation of myostatin, so basically eliminated the production of the protein. And we published a number of papers on that in normal mice and in the dystrophic context. Considering that when you do this in a dystrophic environment, you have to be careful, of course, because if you really increase the muscle mass, you increase the muscle size, but the muscle is not protected by dystrophy, then you actually potentially worsen the disease. And this is something that was noticed and was one of the worrying when there was a clinical trial using antibodies for myostatin for Duchenne that indeed were not at the end working and approved. So the basic thing is that you have to do both treatments. You have to rescue the dystrophin expression, and you can then also downregulate myostatin, so having potentially a more a stronger muscle and potentially even larger muscle. More recently, we have also I would say explore other antisense strategies. This was done with Linda Popplewell and was part of the PhD thesis of Jessica Trundle. And basically, Jess has done this periostin exon skipping. We know that periostin is a fibrotic, is a profibrotic marker, but the version without exon 17 is actually non-fibrotic. So the skipping of exon 17 of periostin potentially can be a system to reduce fibrosis. And we've done this study in this mouse model, which is an MDX mouse, so a mouse with muscular dystrophy. As a model, of course, so it's not really a muscular dystrophy, but it's a very nice model, and it is more fibrotic than the usual MDX mouse because it's in a different background. So this in this case, systemic injection of the oligo basically reduce the amount of periostin with exon 17 and reduce fibrotic markers and improve the grip strength. So this was published, I think, 1 year ago, last year. So there are challenges, of course, for the exon skipping. So we know that the current chemistries are quite inefficient, and we need to use high doses. There is definitely the need of develop new chemistries or new cell-penetrating peptides, and potentially we think that also using synergistic treatments or additive treatments, so for example, antifibrotic approaches or systems to increase the muscle strength may actually help the exon skipping approach. But not only the exon skipping, also the other approaches that are able to rescue dystrophy. And this is the other one that I wanted to talk today. So this is the gene therapy delivery of microdystrophy. And of course, you will be very familiar with what gene therapy does. So this is an in vivo gene therapy. So you really deliver. The aim is to deliver a viral vector expressing the gene, or better, the protein in the bloodstream of the individual, and then having the protein expressing the skeletal muscle. As a vector of choice for this kind of application, it is used generally AAV vector, so adeno-associated viral vector. It has many advantages and some disadvantages. So the advantages are that it is relatively safe, non-pathogenic. The genome does not integrate, which is not true actually, but so you know that it's true that it doesn't integrate as many other vectors do, but there is some integration. Is low. It has a low immunogenicity, again, compared to other vectors, but definitely it has a very efficient gene transfer, that's for sure. You can also choose between different serotypes, and so that you can target specifically certain organs or tissues. Disadvantages are there anyway. So there is a potential seropositivity in human population. So we know that for certain serotypes and the one that are used, sorry, the one that are used for muscle in particular, so, generally AV8, AV9, AV6. For this kind of serotypes, you have 30, 40% of population that is already seropositive. So it couldn't be treated anyway. And this is true also for the DMD population. There is also the need of a high titer to get a proper systemic administration. And this is currently a bottleneck. I have to say in the last few years, is less a problem. Of course, it's very expensive, but this can be done. And there are clinical trials using AAV now that prove that. For us, that we are working in a lab and we work with mice, and we generally do basic science. The main limitation is the limited packaging size of the vector, which is 4.7 kb. So we can't really put into this AAV vector the full-length gene for dystrophin expression. And this is why over the years in our lab as well, microdystrophin were developed. So we developed with George Dickson in particular, and when I was there, I collaborated on this initial work in the lab. Basically, we designed this microdystrophin where we maintain the part of the gene that code for the N-terminal domain of the protein and the cysteine-rich domain as well. The final protein has also 4 spectrin-like repeats and 3 hinges domain. The gene is under control of a SPC512 promoter, which is a synthetic skeletal and cardiac muscle specific promoter. So it expresses very well in muscle. The gene, I haven't said that is actually codon optimized. So this means that the primary sequence is modified, but the final protein remains the same. And you have a much higher expression of the protein at the end. Everything here is packaged into an AAV, serotype 8, that, as I said, works very well in muscle. So this product, AAV8-MD1, is basically curative in the MDX mouse. In the gastrocnemius, in the heart, you really have lots of dystrophin expression. In the diaphragm as well, although a bit less. But you have rescue of muscle force, nearly complete rescue of resistance to central contraction. So in the mouse model, it works very, very well. And so we moved to testing the vector in dogs. So a collaboration with a number of institutes here in France, Geneton Institute of Myology and Atlantic Gene Therapies, that have the colony. It is a golden retriever muscular dystrophy dog with the mutation of the dystrophin gene. And the vector was provided by systemic administration, and the data were actually published here. I'll just show you just a very simple analysis that was done. This was a clinical monitoring made by vets. So every week they were measuring dysphagia, breathing, muscle firmness, and activity of the dogs. And if we consider 100% the clinical score of the normal dogs, the dogs with the muscular dystrophy go down very quickly. And generally, these dogs die when they are 7, 8 months of age. So there's some heterogeneity in these colonies, but most of them really have a very, very severe muscular dystrophy. So the administration of the vector by the low dose actually slightly improved the life of this dog. But the injection of the high dose of the vector basically stabilized the pathology. Some of the dogs were euthanized to do the study. 2 actually survived for a very long time. I know that one of them is still alive after, I think, 10 years from the treatment, so which is incredible. But the study basically went very well, and it was at the basis then of the clinical trial that started in UK and France. This was made by Geneton as a trial sponsor initially. and is based on a single intravenous infusion. And there are, of course, is a dose escalation study, including the therapeutic dose with primary and secondary outcomes. And as a secondary outcomes, it's not just dystrophin levels, but also functional tests. And Genethos has released a couple of press releases recently. This is November last year, that describe how the clinical trial is going. So, of course, the injection have started, and we know now that the first patients are responding very well. So the clinical trial is going pretty well, and the dosing is beneficial to the patients. Of course, in my lab, we don't work with patients. We work with mice. We do preclinical studies. So for us, the vector is very important and interesting, because it allows us to test, for example, other regulatory elements like other enhancer. For example, this is a study that we published in 2021, where we use this CSRM4 enhancer that worked very well with the SPC5B12 promoter to increase the expression of the transgene, in particular in the heart. This is another study that we published, I think, a couple of years ago, again, in collaboration with Linda Popplewell. And this was part of the thesis of Victoria. And in this case, we challenged the vector in a different mouse model. Again, we use this more fibrotic mouse model of disease. And again, the use of this vector basically rescue the specific force, improve the resistance to centric contraction, and decrease the fibrosis in the mouse. So is a pretty good vector. AAV gene therapy is a very promising system, and in US there is now Elevidys, which is one AAV gene therapy approach, FDA approved. But unfortunately, we know that it's not really perfect. So there are data now coming from clinical trials, from even children treated with gene therapy, with this gene therapy application. They show that the stabilization is okay, but will not last forever. This, for example, is Connor Curran. This boy is one of the first children treated with AAV gene therapy, and he did very well from start, responding very well to the treatment. But now we see that the functionality of the treatment is actually fading, and he will probably have to go back to a wheelchair, unfortunately. And suggesting that the therapy can buy some years to these boys, which is fantastic, but it's still not a treatment for a proper treatment for them. For their life. So there is basically two main points here to resolve. One would be the possibility to deliver full-length dystrophin gene. So can we do that? Probably we have to try to do that, but we have to change the vector, which won't be very easy because other vectors bring other problems. Or alternatively, try to be able to do a repeated AAV delivery. That's the other really important point. So I think that there are two crucial points to try to resolve the problem of Duchenne muscular dystrophy. The other disease I wanted to talk is called Oculopharyngeal muscular dystrophy. And this is a different disease. As I said, this is an autosomal dominant genetic disease. It's pretty rare, but it's not that rare in some specific population, like the buccal population, and this is due to founder effects, really. Also in Canada or in Mexico, there are some large population with this disease. And it's a late onset disease, as I said, 50 to 60 years of age, low progression, and there are some primary muscles that are affected. Particular muscle of the eyelid, so you have something called eyelid drooping, so ptosis, and the muscle of the cricopharyngeal area, and there's swallowing difficulties for these patients. But however, late in life, all muscles are basically affected, and the patients have to use a wheelchair later in life. So it's true that there are the primary muscles that are affected, but not only. The disease is due to expansion of the GCG trinucleotide repeat in a gene that code for poly-A binding protein nuclear 1, or PAPPN1. It's a protein that has lots of different roles. I just put 3 here. Importantly, is involved in the stability of the RNA messenger, and also in the choice, the usage of the alternative poly-A sites in the messenger. So it's very important. It's very important protein. And the fact that there are these expansion GCG trinuclear repeats, it means that you have between 11 and 17 alanines instead of the normal 10 at the end terminal of the protein. And this makes a protein that aggregates, basically. It misfold, aggregates, form intranuclear inclusions. So this is why we have a loss in gain of function disease. So we have a gain of function disease because we have the aggregates. We have a loss of function disease because you have less of PAPIN-1 that can work in the cell. To study the disease, we use this mouse model, which is a great model. It's called A17 because it has 17 alanines in its PAPIN-1. But actually, this is a transgenic mouse. Is generated using expanded bovine PAPIN-1 with 17 alanines. It is overexpressed by using human skeletal actin promoter. So it has basically the normal murine genes, plus it overexpressed this bovine version of mutated PAPIN-1. And is very nice model, all skeletal muscle containing intranuclear inclusion that you see here in green, for example. There's fibrosis, muscle atrophy, gene deregulation, muscle weakness. So it is a great model to study the disease because it really recapitulates. There's a possibility, and this has been tested by different groups, including us, to try a pharmacological approach. So try to decrease the amount of aggregates. And we have done this and published as well using a drug called guanabenzacetate. And we saw that it actually worked, but not really as other systems. We tested antibodies for myostatin. As I mentioned before, there is this system to downregulate myostatin and so increase the muscle size. In this disease, you have muscle atrophy. So in theory, that could help. And indeed, in the mouse model, it helped very much. But again, gene therapy is the system that we think is the best option here, and that was developed in the lab. If you consider that this is the genotype of a patient, patients are generally heterozygous for the mutation. So one allele is normal, the other allele has the mutation. Because of the type of mutation we have, so trinucleotide repeats, we can't really target a specific allele. So the only way to try to find a treatment for this disease is actually a silence and replace approach, where you suppress both the expression of the alleles. And so all the endogenous PAPIN-1 is suppressed, the normal and the mutated, and you replace the function of the normal version of PAPIN-1, providing another working copy. So we have done that using 2 vectors initially. One vector with 3 shRNA, the short RNA, targeting all endogenous PAPIN-1 to the cell, and providing a codon-optimized human PAPIN-1, which was resistant to the cleavage by this shRNA because it is codon-optimized. So the primary sequence is different from the sequence you have, the endogenous PAPIN-1. So the silence and replace approach was tested in the mouse model, and again, we saw a normalization of the specific force, for example, in the mice treated with the 2 vectors together compared to the FEB, which is the wild type. And we also saw a strong improvement in transcriptomics. So if you consider we have 452 upregulated genes in the A17 compared to the FEB, the different treatments, so either the single vector or the 2 vectors together, tend to decrease the expression of the genes, with the best condition being actually the 2 vectors together, and the opposite for the genes that are downregulated in A17 compared to the FEB. So at the end, with the treatment, with the gene therapy treatment, you have a 98% of correction of the transcriptome. And all the data were published in 2017. And this allowed to move from a two-vector system to a single vector system in collaboration with the biopharma company, and where basically we have a different approach here, because we have a single promoter, a SPC512 promoter, skeletal and cardiac muscle specific promoter that transcribe a codon-optimized wild-type PAPIN1, and also two shRNA that are embedded in microRNA backbone. And then they are processed in different ways. So basically, you have the RNA messenger here is processed in a way. Here you have the generation of microRNA or siRNA, I would say, that actually have the role of downregulating the endogenous PAPIN1. This vector is called BB301. Is into an AAV9 modified. But what is great with this vector is that his administration is really dose specific. So if you increase the amount of the vector, you really see a decrease in the number of intranuclear inclusions, up to 0, more or less. And if you increase the amount of the vector, you normalize the muscle weight to the level of the FEB. And the same effect you see with the muscle force generated by the mouse. And also the knockdown of endogenous PAPIN1 is dose dependent. Again, if you increase the dose, you have an increased downregulation of endogenous PAPIN1, increase upregulation of the SH-mirs, and increase upregulation of the codon-optimized PAPIN1. So all this data were actually part of this publication. And that was very important because then allowed the further studies. This is a study that was done in the sheep. And in this case, basically, the sheep was chosen because it has an anatomy that is very similar to the human one. And in order to understand where to inject the vector, that was a very important study to be done. And this is because in this case, we are talking about a local administration of the gene therapy application, specifically in the cricopharyngeal muscle. After that, when the initial study in the sheep was done, other studies were performed in these bigger dogs. They are not model of oculopharyngeal muscular dystrophy because there are no large model of the disease. They are just wild-type animals that have been used to understand how to perform the delivery of the vector. So that allowed to start a clinical trial that is now ongoing. This again is a press release of last year. They showed that the first patient injected with the vector is as a benefit, and there were lots of analyses that were performed on that, but it's only one patient. So suggest that we have to be optimistic, but of course we will have to see the other patients how they behave. So the injection of BB301 is promising as a local gene therapy treatment, but of course OPMD patients has also impaired mobility. So as a lab, we are particularly interested on develop a systemic treatment for OPMD. And of course, there's a problem here that the current vector is made using shRNA, which have a number of problems. So for a local administration, they are certainly fantastic, but for a systemic administration, there are potentially some issues. First of all, the overloading of the microRNA machinery pathways is a big problem. The fact that there can be off-target effects, in particular because when you deliver these shRNAs, you don't really deliver a single sequence. You deliver a family of a sequence when there are overhangs in respect of the oligo you believe you are actually delivering. So there are potentially off-target effects there, and of course with the NAAV delivering this shRNA, you have the problem that you permanently switch on a system. So you can't really stop it there. So we are now trying to improve the gene therapy vector. So we are doing many things to do it actually, but actually just a couple. This is a work of James Marsh in my lab. So, for example, we are changing the promoter. We are also, as shown here, try to find a new codon-optimized PAPN1 sequence that can be better than the one we have in the current vector. Here we have this, but yeah, but this is actually measured consider the PAPN1. This is a flag because for our study, the PAPN1 we have used was linked to a flag, but they clearly similar expression. And what you see is that there are codon-optimized PAPN1, like this, for example, that actually behave better than the sequence-optimized PAPN1 that is currently in the vector. And also, in the vector, we have two shRNA called 13 and 17. And actually, we could find other shRNA that act individually better or at the same level than when these two are put in pair. So potentially we could have a single shRNA instead of having two, which reduce clearly the off-target effect. So conclusion, gene therapy treatment in mouse rescue the pathology pretty well. This allowed to have the experiment on a larger animal model that indicates that intramuscular delivery is also possible, and it's not trivial actually, because if you consider that you have to inject a large muscle, it's not like injecting a tibialis anterior of a mouse. In this case, you really inject a large muscle that is not trivial. This basically means that the phase 1-2 clinical trial was possible, and preliminary data are encouraging. But of course, we need other steps to improve the vector to get to systemic administration. And I stop here. I just wanted to acknowledge the people in the lab, so James, Alexis, and my two PhD students, Ru, Herida, and the lab manager, Penelope, and the collaborators and the founders of my research. And I thank you for your attention.
Hi, Alberto. Thank you for that fantastic talk. I think there's a lot going on in the field currently, which is very exciting. We don't have time for too many questions, but I will bring one from the audience. So Ellie from UCL asks, out of all the possible strategies that we touched upon, which one do you think is the most exciting moving forwards now that the gene therapy field is maturing?
Yeah, I think that I think personally, I think the AAV gene therapy application is going to be very exciting in the future. I think that there are many groups now that are not really focusing on the AAV microdystrophin, for example, itself, but all the other problems that are correlated to the delivery. So, for example, the possibility to re-inject the vector, and of course, it will involve many people working on immunology, for example. If we can combine the knowledge on immunology and molecular biology to create better vector, potentially better serotypes, modification of the serotype, system to shield the vector from the immune response. So things like that can actually allow the re-administration of the vector. That would be very, very exciting, I think. But of course, also exon skipping system to improve the exon skipping. So all of these techniques are potentially very promising and can be improved.
Yeah, I agree. I think re-administration is a big topic in the field. I do have one question. I thought the suggesting, combination of therapies was very interesting. Do you think, particularly for DMD, some of these therapies, for example, the use involving myostatin or periostin, will be something that could potentially move to clinical trials in the future, combining certain
I think
therapies?
yeah, potentially. I think they need to be improved. myostatin in particular has been in clinical trial. As I mentioned, there were antibodies that were tested. The problem is that antibodies as well have they are potentially toxic. So you need to have better antibodies that can be tested. They need to be properly provided. So you have also to have a system to rescue dystrophin, at least in the Duchenne muscle. Otherwise, you may not see really an effect. So there are different. There will be different issues associated to the other therapies that you want to combine with the one rescue dystrophy in the muscle. So if you can have a proper system that works fine, definitely they can be combined. I think it won't be immediately; it will come later, and we always have to consider that currently the patients that are taking AAV gene therapy, are taking exon skipping, are already under a combined therapy because they are also under steroids that provide other effects. So there is already a combined therapy there. So the combination will for sure be the future for the treatment of these pathologies in muscle.
Well, thank you so much for your time today and for this amazing talk. It's all very exciting work, and hopefully we'll have you again at some point in the future.
Thank you, Rebecca. Thank you.